Okay, so for this audio lecture, we're going to be covering chapter 13, so looking at microbe-human interactions. So we want to keep in mind that humans can act as a host for microbes, and so our body surface is constantly being exposed to microbes, like from air, from soil, from other things that we touch. And on the most part, most of us are healthy on a given day. And so in the rare case, it would actually lead to an infection, and that depends upon us as the host and the microbe that we're interacting with. Okay, so we all are walking around with subclinical infections. There's microbes present, but we're not showing any signs or symptoms of the infection. Okay, an infection would be where we're showing signs and symptoms of it. With pathology, we can have a pathological state where because of the infection, there's some kind of damage or disruption to tissues and organ systems in how they normally would function. So if you think about A&P, you learned about an organ system and homeostasis and how it normally was. If there's microbes growing in there to high numbers where you have an infection, it wouldn't be able to function the same. So again, some of these signs and symptoms we would see because of these disruptions. When we have an infectious disease, that's when it is caused by microbes or their products, because some microbes can produce toxins that the microbe itself is not actually disrupting the tissues, it's the toxins they release. So we all have resident microbiota, and if we look at us, what you would consider you, and we looked at individual cells, you're actually more microbial than you are human. So it's estimated that for every one human cell, you actually have 10 microbial cells, and that'd be bacteria and yeast on you. If we look at genetic material, it's even further skewed, so that for like each nucleic acid you have from your human cells, you actually have like 100 nucleic acids from microbes. So the Human Microbiome Project, similar, most people know about the Genome Project, looking to sequence the genome. The Human Microbiome Project looked to see what were the normal resident microbes that were on humans. And so there's definitely work coming out all the time from the Human Microbiome Project, but also other projects that have kind of offshot from that. So one of the papers I love is this paper that was published in PeerJ. So it actually looks at the idea of this microbial cloud. So again, we think of ourselves as human, but we actually have this and when we think of residents, we think of it being attached to us. But the reality is we actually have this microbial cloud around us. So we're a little bit more like Pigpen. So if you're familiar with the cartoon, where Pigpen's on it, wherever Pigpen goes, there's this like dust cloud around him. And so that's the reality of what we are. So this study, what they did was they had the subjects were sitting at a desk that was in a sterile room. Desk was sterile. They had a computer so they could binge watch Netflix, and they had an air filter that was above them, so that if there was material that was not tightly attached to them, it would actually get caught in the filter. And what you're looking at here is that of these subjects, you'll notice that none of these subjects look the same. They're a little bar that they're made because this is looking at that genetic material from microbes. And you can see that each one of these, the bar looks different. Some have more diversity, meaning the bar is longer. It's found in different locations. And so what ends up happening is that your microbial cloud is unique to you. So it actually can almost be used as a fingerprint to identify you. And there is an area of criminal investigation that's actually looking at utilizing microbes. So microbes that might get left at the crime scene. And would this be able to actually identify who the suspect was if no actual fingerprints were left? And this is definitely used looking at trace materials, like spores that are present to indicate whether a body has been moved from one location to another. So this is not really so they've looked at fungal spores and certain fungal spores and pollen is unique to a certain location. So it's not too far out there to think that in the future, we would have our microbial fingerprints, and that might get us in trouble, even though we think of cleaning up, all the hair and fingerprints we might leave behind. So some of the terms used with microbiota are the resident microbes. So again, these are the ones that kind of are ours, unique, but they do change over time. And again, they become like a fingerprint, and they tell the story of our lives. So if you're a person who travels a lot, you're going to have microbes that become your resident from all over the places you travel or from what you eat. So the resident microbes on someone who consumes meat is different than the resident microbes for someone who's vegan or vegetarian, or someone who consumes less of alcohol. And so again, all of those things we do, from diet, from exercise, from travel, from where we work, all of that ends up shaping our resident microbes. We then have transient microbes. These are the ones that don't normally reside, they kind of are passing through. So we might pick them up when we touch a door handle, or we borrow a pencil from somebody, but they're not, we have all those residents there, so they don't get strongly attached, so they tend to then get dropped off at the next door handle we touch. So most of these are harmless because, again, on a given day, most of us are healthy, but there are some that are pathogenic. So they're pathogens, so they can cause disease. We also have opportunistic bacteria and microbes, and these are, if the conditions are right, they will actually cause illness. So normally they could be living with us in a very symbiotic relationship. They could be our resident or transient, but if there's a breakdown in our immunity, either because we're taking immunosuppressive drugs, or maybe we're not sleeping enough, or stressed out, or there's certain medical treatments that are done, or you have an implantation of a device, so you have surgery done, that can introduce these opportunistic bacteria to a different location. So if we have skin bacteria and it moves into the abdominal cavity, this changes its environment, and so it can then exhibit pathogenic behavior and cause an infection. Where if it had just stayed in the skin, it never would have done that. Okay, so there's an area of research that's looking at these opportunistic microbes and part of this research, or this is one paper that's really interesting because it looks at influenza and how it can change the behavior of microbes. And so we know that individuals who have influenza have a higher risk of secondary infection with strep. And so this paper actually looks at how influenza disrupts these biofilms that the strep is in, and then that kicks the strep out, and now it has to go fend for itself. It's lost all its friends in the biofilm, and it's really pissed off, and that's then can cause strep pneumonia. So you can get pneumonia from that, and it's really aggressive forms of pneumonia for individuals who end up in the ICU or die. So it's not like it behaves very differently than your classic strep pneumonia. So when we look at the normal microbiota, through the Human Microbiome Project and additional research, we know that there are many locations that normal microbiota live on, definitely in our skin, respiratory tract, our GI. So the role of your microbiota in your GI tract is, exploding in the area as far as an area of research, but also in the reproductive tract, urinary tracts too. And the reality, and I'll show a slide in a moment, but the reality is your normal microbiota really is everywhere. So this is just showing you some of the different normal microbiota that might be present. So in panel A, you can see that there's these orange spears that are there, and that this is actually from your nose. So these are maybe like Staphylococcus or Streptococcus that are present. In panel B, these are bacillus that would be present in the lining of your stomach, so these could be like E. coli. You can see in the large intestine this mixed population. So when we look at normal flora, it can have several different impacts on us as being the host, humans. So I think of 3 impacts of normal flora. Okay, so the first impact is that it takes up space. So it actually prevents pathogens from attaching because it's there. So it's just this exclusion, not allowing for pathogens to be able to attach. So think of like location, location, location. So if you have your surfaces inside and out covered with beneficial, happy microbes, you can't get those angry, mad microbes that cause disease to attach. Also, your normal flora plays an important role in your metabolism. So those microbes that are in your gut, they're producing digestive enzymes. So we saw in our last audio lecture how bacteria produce digestive enzymes to break down material. So yes, your digestive system is amazing. Stomach, whoa, we're so proud of our stomach and the acids and digestive enzymes it produces. But those microbes in your GI tract are also breaking down the food you eat. They're doing it for themselves, but you also benefit because there's more nutrition there for you. Okay, so you can get like more bang for your buck for the food you're eating. Now, obviously, you know, if you're eating unhealthy, your microbes will actually produce hormones that go through the gut-brain axis, and they'll tell you to eat more of that junk food because they like the junk food too. And so when we have bad eating habits, is it really us or is it our microbes? And so we can actually shift our microbes in our gut by changing what we eat, right? And then it changes what we want. And again, is it us or is it the microbes? Okay, so metabolism is really important. They also, besides the digestive enzymes, they also will produce vitamins. So E. coli in your gut, beneficial E. coli in your gut, will produce vitamin K for you. So again, it's a really symbiotic relationship because the E. coli needs vitamin K, produces excess, and you benefit from that. And then as an immunologist, last but not least, is how your normal flora actually shapes your immune system. So from very early on, your normal flora will end up helping your immune system recognize what it should respond to and what it should not respond to. Okay, and so how we acquire our normal flora really shapes that education of the immune system. And we'll talk more about that in our next slide. And it'll probably later on when we talk about the immune response, we'll talk about later on in the semester when we talk about the immune response, we'll end up talking about how your normal flora, your friends, actually will shape the response that you get. So if we look at how we acquire our normal microbiota, it starts from day 1. So there's actually some microbes that can be crossed through the placenta. So definitely in utero, there is some shaping of the microbiota, but the first major event of acquiring normal microbiota would be birth. So if you were born vaginally through the birthing canal, microbes were deposited on you, on baby, okay? And so now those microbes are coming from the mother's vaginal cavity. So a lot of those are going to be like lactobacillus bacteria, which are the same bacteria that are in yogurt. Okay, not to gross anyone out, nobody will be able to look at yogurt quite the same way. But then what food baby eats. So there are bacteria that are going to be passed through breast milk. So there's actually gut microbes from mom that actually migrate and move into the breast tissue, into the glands, and actually get secreted in breast milk for babies who are breastfed. So they're acquiring gut microbes, their normal flora, through breastfeeding. And then just the simple act of breathing, we're going to acquire our respiratory microbes. Eventually, baby's going to start moving around, put their hands and feet into their mouths, and so what ends up then they're picking up is coliforms, and eventually end up picking up anaerobic bacteria. But if we think about lactobacillus, these first bacteria that are going to be present, these are pretty benign. I've never heard of anyone, dying from a lactobacillus infection because people eat yogurt every day. And if there's an issue with yogurt, it's because there's some kind of contaminant. It's not the lactobacillus bacteria. Okay, so these are the great first friends for your immune system to recognize. Well, I don't have to respond to every single microbe. That's a fine microbe. And then the coliforms come in. So these would be a lot of these gut microbes. Some of them are gram-negative bacteria. And so, again, those are going to be around your whole life. So we want to make sure we're not responding to those. And definitely not responding to the strains that are not pathogenic. Then we get these anaerobes, and these are more potential pathogens. And so now your immune system knows, okay, well, we know not to recognize these certain bacteria, right? And so these are the ones we need to actually respond to. So then we want to think about, well, what happens if this is altered, right? So babies are not always born through vaginal births, so C-sections are needed. So disclaimer, Dr. Schreiner is not saying C-section should not happen because of potential changes to the normal microbiota. What I am saying is we need to be mindful of that, and what can we do to correct that? So babies who are born through C-section do have potential for having higher incidence of allergy, asthma, autoimmune disorders. So these would be disorders of the immune system. And so the idea is that because there's been a shift in the microbes that first populated, because they wouldn't have those vaginal bacteria present, those lactobacillus, that this actually causes an altered immune response in them. Okay, there's some newer literature out that's showing that there's actually this, at 1 month, there's this kind of like retraining of the immune system, and that might be a pinpoint where we'd want to look at babies who are born through C-section, and is there a way to take advantage of this kind of dramatic shift in the immune system at 1 month, that we could use that to our advantage, okay? So again, not saying that we shouldn't have C-sections, we just need to be mindful of what these changes. And so is there a way to inoculate baby with lactobacillus bacteria right after their birth, so that they're not, their first bacteria they're seeing that's attached to them isn't the skin bacteria from a nurse, or from the doctor, or from another family member, okay? And so there's studies, there's a study out from University of California, Davis, UC Davis, where they were looking at taking mom's vaginal bacteria and wiping baby down with that. So again, kind of mimic the process of vaginal birth, where the bacteria gets populated. So again, another main kind of altered would be formula feeding. Again, I'm not saying babies shouldn't be formula fed. Feed your baby. I don't care how you feed your baby, just feed the baby. But again, we're not getting these gut microbes in. Well, don't worry, formula companies are looking at this, and so Nestle actually has a formula that does have basically probiotics in it, and it's designed more for colicky babies. So colicky babies will get gas production, and so by giving these babies beneficial bacteria, it helps them to break down the material better so they don't get the gas production. And so this is probably something that you'll see right now as a prescription formula, but you'll probably end up seeing going mainstream, where we have these probiotic formulas. And again, it's meant to mimic, to restore those gut microbes that baby would get if they were breastfeeding. So again, once again, Dr. Schreiner's not saying that babies should only come in through vaginal birth or only be breastfed. What I'm saying is that all we need to do is medically think about, when we change this, what do we need to do to replace those normal microbes? So this paper actually looks at some of these altered states that can actually interfere with the development of our normal microbiota. So if we're being exposed to antibiotics, whether we have C-section, whether we're excessively washing our skin with antimicrobials, and also if we're ingesting antimicrobials, those would all affect our normal microbiota. And so again, if we're partaking in these, we need to think about what can we do to prevent that. So obviously, especially in COVID times, it's really important to wash your hands, but we do need to be mindful of making sure we go play in the soil too, so that we're repopulating ourselves with good beneficial microbes. Now in your textbook, there's a table that has sites and fluids thought to be sterile, and I would emphasize thought. So I have to check the newer edition because I'm sure it will be changed. So there's definitely microbes that are found in these tissues and organs that are listed here that are actually beneficial. So they're not sterile. For one, there's living tissues there. And two, there's definitely going to be microbes there. So if you've brushed your teeth recently, when you brush your teeth, you actually get some of your oral bacteria into your bloodstream. So your blood is not sterile. What happens is there's a whole immune response we'll talk about later on called complement. And complement actually removes that bacteria but allows your immune system to see it. So again, reinforcing what's normally there and being able to check whether this is actually a pathogen. So when we look at the microbe-host interaction, as I mentioned, we have to think about the microbe and what is the microbe bringing to these interactions, but also the host. So there's a number of factors that can influence. So it really becomes a perfect storm whether you actually end up getting disease. So if you look in the top box, we have our microbe. So each microbe is going to have a virulence, so how easily it can actually cause disease. And this is due to a whole bunch of virulence factors or pathogenicity factors. So if a bacteria can produce endotoxins or exotoxins, or if they can have fembrii or produce certain enzymes or produce enzymes that block phagocytosis, process we'll talk about later on in the semester. These are all would put this into the high category. And then each microbe is going to have an optimal infection dose, and so depending upon where that is. And then lastly, we need to have the correct portal of entry. So most microbes like a certain way to get into you. So whether they're respiratory or digestive. So if you eat a respiratory microbe, that's not the right portal of entry, so that's in the no switch, and so it's unlikely that from the microbe side, that you would actually end up with disease. Now, if the bottom box, we look at the host, you're going to have different genetic components. You know, how much of a certain immune cell, like a macrophage or dendritic cell, or an eosinophil that you have. So all these non-specific defenses, and so you're born with that. So that switch is kind of stuck there, depending upon who your parents were. And then if we have a previous exposure, that would be specific immunity, so our T cells and B cells, antibodies. And so if you've seen the microbe before, whether naturally or whether because of a vaccine, you would have that in the yes switch. If you haven't been vaccinated, you haven't seen it before, it's in the no switch. And then your general health level. So whether you're producing lots of stress hormones, overtired, eating poorly, that also factors in how you would respond. So again, it becomes this perfect storm from the microbe and also from you as the host, and whether the microbe's just going to pass through and nothing happens, whether it becomes established and you're colonized, and so this is where you're a carrier for it, so you may not actually show symptoms of it. And then where we would actually recognize that you do have the microbe present where you have disease. So again, perfect storm, lots of variations that can happen. And that's why, for the most part, most of us are healthy on a given day. So factors that affect susceptibility would be age, right? So extremely young, like newborns, their immune system hasn't developed, and also elderly. So the older you get, I 30 to 40 is kind of the prime of your immune system, and then it starts to wane. Some of those memory cells you had as a child aren't there anymore, and then some of these cells just don't function as well as you get older, so you have increased susceptibility to infection. We can also have genetic defects, so these you could be born with them, or you could acquire them due to like viral infection. So obviously, if your immune system is not fully functional, you would not be able to respond to a microbe infection as well. Also, if we've had surgery or an organ transplant, and a lot of times that's because we might have been put on to antibiotics or immunosuppressive drugs. So we're kind of in an altered state of how the immune system would act with antibiotic. If we take an antibiotic, we're actually selecting for ones that are resistant, that the antibiotic doesn't work against, so that could increase our susceptibility. Also, if we have certain underlying conditions like cancer, liver disorder, or diabetes, again, these alter our immune response, and so it affect our ability to respond. All right, so if we look at the stages of infection, for this we are looking at the point of view from the microbe. So it has to go through a number of steps in order to even get to the point where it's causing disease. So the first step would be to actually find a portal of entry. And so again, as I mentioned, for most organisms, they're going to have a preferred portal of entry. So whether it's your skin, your GI tract, respiratory tract, your genital tract, or exogenous because it's being moved from one location to another. Okay, once inside, it then has to attach firmly. So this is where it has those fimbriae, the glycocalyx, capsules, slime layers, surface proteins, or viral spikes that allow for attachment. And you then have to survive the host defense. So your host is trying to fight you. So you have to avoid phagocytosis, avoid dying inside of that phagocyte, and block antibodies from binding you. If you've made it successfully past this stage, now you can cause damage, which would be recognized as signs and symptoms disease. And so you can cause direct damage through toxins or enzymes, and then you can also have indirect damage based on the host response. So if the host responds kind of excessively, the host will actually damage tissues for you. And a lot of what you're seeing with COVID is actually indirect damage where the host immune response is causing damage to tissues. Okay, and after that, because you're a good pathogen, you want to find a new host, so you need to exit. So you have a portal of exit. So a lot of times these portal of exits kind of go with the portal of entry. So if you came in and used the GI tract, you're going to exit through the GI tract. If you're a respiratory and you came in through an inhale, you're going to be released through an exhale. But we have other portal of exits like blood also. So just looking at each of these steps a little closer, with portal of entry, again, it's how the microbe gets in. The most popular portal of entry is respiratory, so if we could just stop breathing, we would diminish the number of infections dramatically. So obviously that's not an option, and this is where filtering or protecting the respiratory tract, protecting what's entering, can actually help to minimize or prevent illness. Okay. And then with attachment, in here you can see the bacteria with its fimbriae attaching, or the formation of this biofilm that allows it to attach through the use of its glycocalyx, the capsule, or the slime layer. And then we have our virus that's using its spikes to attach to the host cell. So there are a number of different attachment molecules. So this table just outlines some of the different microbes, the disease they cause, and how they attach. You'll notice in the top part section of this, most of these are fimbriae that allow for attachment. Then towards the bottom where we have our viruses, it's going to be those viral spikes that allow for attachment. Okay, so I would say, if you have your favorite disease or favorite organism, see if it's on the chart. But again, you should recognize that there's certain structures like fembrii and viral spikes that allow it. I will point out for cavities, we have Streptococcus mutinus, and so it actually has the slime layer that actually allows for attachment. And so that's why, if it's been a while since you brushed your teeth, you might feel that kind of slimy layer. So step 3 is surviving the host defense, and so a big part of the host defense will be phagocytes. These are white blood cells that undergo the process of phagocytosis, where they engulf and destroy pathogens and foreign material and damaged cells. And so for phagocytes, the way to stop them is by producing anti-phagocytic factors. And so microbes can produce leukocidins. Those are toxins that kill white blood cells. Or they could have an extracellular surface layer that makes it tough for them to attach. Again, that's where the capsule layer, the capsule form of the glycocalyx would help with that. But then we can also have it so that they don't allow the digestive enzymes that kill the pathogens in phagocytosis to actually do that. So there are microbes like rickettsias that actually can live inside of the phagocytes. So they actually will grow and divide and increase in numbers. And eventually they can actually cause the phagocyte to burst open and they're released. So it's pretty cool strategy in order to survive. So then looking at causing disease, this is where we can get either direct damage, where we see these exoenzymes or toxins being produced by the bacteria that damages those cells that make up the tissues. But at the same time, we can have this over-response by the immune system that can cause damage. So we're going to look a little bit more at endotoxins and exotoxins. So gram-negative bacteria can't have endotoxins, endo within, exo out of, secreted. And so again, endotoxins are something we see in gram-negative bacteria because it's LPS. So if you look at chemical compositions, lipid polysaccharide, LPS, and it's not really the cell wall, it's the outer membrane. So remember, gram-negative bacteria have that outer membrane that have LPS in it, and LPS has different subunits to it, and so the lipid A can get released, and that can get incorporated into any cell. And so this is why if we look at effects on the body, it has a systemic effect. So we would have fever and inflammation occurring. The good thing, if there's a good thing, is that it has to be at a high dose. So if we look at toxicity for endotoxin, we need to have a large amount of it. So normally if our immune system is good, is working well, we'll keep those gram-negative bacteria at lower levels. So just to point out on the manner of release for endotoxins, again, it's going to be released by cells when they're lysed or killed. So it's not the gram-negative bacteria is alive, it's been killed, but lipid A ends up getting released. When we look at exotoxins, these tend to be gram-positive. There are gram-negatives that do produce exotoxins, and looking at the manner of release, they're going to be secreted from live cells. So the cell has to be alive, so if you've killed the cell either by the immune system or by an antibiotic, they cannot produce exotoxins. Okay? These, if you look at the chemical composition, are small proteins or peptides that are produced. So again, cell has to be alive to undergo protein synthesis to produce the exotoxin. So again, if you kill it, yay, you can't produce exotoxins. Also, because these are protein-based, you can actually have antitoxins, so you can actually produce antibodies to it, but also medically we have neutralizing components that can actually work as antitoxins against it. So if you look under immune response, you can see how exotoxins are different than endotoxins. Another important characteristic of an exotoxin, because they're proteins, they are unstable. So if we have a fever, they tend to denature, so they would not work against that. Also, these exotoxins are going to be specific to a cell type. So the cell has to have a receptor to bind that exotoxin, that protein, in order for it to have an effect on the cell. And not all cells will have a receptor for it. So these exotoxins become very specific to a certain organ, tissue, okay? But because of that, you only need a small amount of them in order to get the effect, okay? So definitely one of the things you should be able to do is compare and contrast endotoxins versus exotoxins, be able to pick out some of those key characteristics of them, so that, you could answer a short answer question or answer some multiple choice questions on them. So this figure just is showing how those exotoxins are going to work on a specific or target organ or tissue. So an exotoxin being produced could affect the cardiac tissue, or it could affect lung, or it could affect liver or GI, but it's not going to affect all. Where with endotoxins, they get incorporated into cells that have cell membranes, which is every cell, and so that lipid could become detrimental. So you end up with a systemic effect. Okay? And again, it's being released from cells that have been killed. When we look at types of infection, we can have localized infections that are found in one location, sometimes referred to as boils, or we can have a systemic infection that would be throughout the body. Okay. Many infections are actually mixed infections, and so this is where, as we get more powerful techniques in the lab, we can actually identify some of these smaller populations that are present within an infection. And so some of these infections that we previously thought were because of a single organism, like acne, there's P. acnes, this poor bacteria got its name based off of the fact it was isolated from some acne way back when. But the reality is that it's actually a mixed infection. So Staphylococcus aureus has to be present to produce acid. That acid creates an environment that P. acnes likes, and so it's really this combination of the two together. And if you treat for the Staphylococcus aureus, you don't get the acid production, so you don't get the P. acnes, so you don't get acne. So again, as research is coming out, we're finding more about the complexity of some of these infections. We can also have where we have a primary infection, and because of antibiotics taken or kind of shifting of the immune system, we can end up developing a secondary infection. So secondary infections are, sometimes a complication of certain drug treatments or treating a primary infection. And again, lastly, we have that portal of exit. So we need to get if we're going to be a good pathogen, we have to get out of our host to find a new host. And again, sometimes, as everyone's aware with COVID, through coughing, sneezing, and just breathing, we can actually exhale pathogens. But the same is true with, has been true with like flu and other respiratory illnesses. With GI illnesses, usually it's through fecal or urine output that the microbe moves onto its next host. Now, looking at from the other point of view, looking at the point of view from you as the host, when there's a microbe present, when there's a pathogen present, you're going to go through a progression of infection. And so first we're going to have this incubation period. So that's where the microbe is increasing in numbers. Your immune system quietly is, fighting it and trying to control it. But eventually you might move into the prodromal stage. That's when you're not feeling quite right. You're not 100%. You're not sick yet, but you're maybe you feel a little tired. Maybe there's a little scratchy in your throat, but not like a full-on sore throat. Eventually you move into the period of invasion, and this is where your signs and symptoms increase in severity. You get to the point where you have the height of infection. this is it can't get any worse, just make it go away. And this is where your immune system is in like full gear. Your adaptive or specific immunity, your T cells and B cells are now activated in trying to clear that infection. And then we move into the convalescent period, and this is where you are your immune system is now clearing out those microbes so that you can begin recovery. There can be chronic carriers, and chronic carriers could be ones that go through this period of infection and recovery, but they don't clear all of the pathogen, and so they can either go through chronic infections, or they can go and become chronic carriers in which they're spreading. And the reality is that in many viral infections and even bacterial infections, we actually can be spreading the microorganism before we are actually in the period of invasion. During the period of invasion, usually this is where we call in sick. But even in the prodromal stage, this is where you could start to actually spread. So this is where there's this window where you're an asymptomatic carrier, pretty much for any illness. Really come into attention with COVID. And so generally 2 days before you start to show symptoms, you can actually spread COVID. And again, there could be individuals who never develop symptoms but could be spreading and could even be super spreaders of COVID. So, this graph should look somewhat familiar. So last audio lecture, we talked about growth curves. And so this really overlays with the growth curve so that during the lag phase is where you, the host, is experiencing the incubation phase. The beginning of the exponential growth phase is where we have prodromal, and then as the bacteria moves through, or the microbe moves through exponential growth phase, we are moving into the period of invasion, and then we go into stationary phase where we're at the height of an infection, and then the death phase would correspond with convalescent. There are a number of signs and symptoms when we look at infectious diseases. Signs are going to be something that we can measure. So we can know whether you have a fever because we put a thermometer, we take your temperature, or we can take a blood sample and run a CBC and see what your levels of leukocytes are. We can palpate for swollen lymph nodes. We can test for antibodies. We can listen to your heart to see whether you have tachycardia. So again, all of these would be signs, things that someone can assess and measure. Symptoms are up to the patient to report. So, are you chilly? And so normally when we're face to face, I would talk about how we could talk about, is this room the right temperature or not? Are you guys cold? Are you warm? And everyone would come up with a different opinion, right? We can think about a painful event in our life, a physical pain. And again, everyone's going to have a different experience. I dislocated my clavicle. I have my patella slipped. Those were not painful events for me. I gave birth to 2 children, not painful to me. Most painful experience is going to the dentist for a routine cleaning. Okay, so I obviously understand there's a lot of psychological things going into that, but again, everyone has a different pain threshold. And if we think about fatigue, there are people who are tired all the time, and then there's people who never feel fatigued. So when we look at symptoms, it's really, there's a lot of things going into that, and that can vary from patient to patient. And so symptoms aren't always the best indicator to use clinically. Signs where we can measure those or visually observe it are better to indicate infectious disease. Obviously, we do have to rely upon symptoms, but again, we want to keep that in mind. Also, we have to keep in mind that some populations wouldn't be able to communicate their symptoms. So small children aren't going to be able to communicate, they're not going to understand certain aspects, right? So again, for them to be able to communicate that. We also have certain populations that may not be able to communicate, either because they're mute or because we don't speak the same dialect or language. Also, you could have someone who's unconscious, right? And so you can't rely upon symptoms at that point. You'd have to rely upon signs anyways. So again, all these different, sorry, froggy there. So again, all these different factors play into the role whether the microbe will be successful at causing disease. And again, keep in mind, not all microbes are trying to cause disease. Some are just trying to live symbiotic lives with you. But there are some that, because of the genes they have present, they are more aggressive, they are more pathogenic, and they're going to be more likely to cause disease. So pathogens can persist in reservoirs. These are primary habitats in the natural world for pathogens. So we can have biotic reservoirs or living reservoirs. So this would be like another mammal or, bird, tick, mosquito. So again, living. Or we could have an abiotic reservoir, which is non-living. So that would be like a rock. Okay. In that, most textbooks include soil and water in there. And so water at its root of H2O, yes. But if we think about a stream, there's a lot of other additional components in there, but that's non-living. And then soil in itself has microbes present in it. So I always have a tough time when textbooks say soil, because when as a microbiologist, when I think about soil, I think of all of the different organisms in there, and you would never be able to find soil that doesn't have all these organisms in it, because then it's not soil. But that's a whole different point. So for examples of abiotic, non-living reservoirs, like a rock, soil, and water are acceptable answers, even though I struggle with those. So as I mentioned, we can have asymptomatic carriers. So we can have individuals that have the microbes present, not showing symptoms, but they are actually shedding the microorganism, so that we then could pick it up. We would have that incubation, and then during that incubation period, we could be actually shedding those microbes also. And then during the convalescent period, even though we're feeling better, we could also be shedding. And that's why a lot of times, employers will say you need to be 24 hours free of a fever, 24 to 48 hours free of a fever with no medication on board. And that's just because during that time you could actually be shedding. And then again, we can have these chronic carriers who, even though they're fine, they haven't had a fever, they could still be shedding pathogens. Okay. The transmission can be passive. So this is where fomites, material that gets contaminated with the pathogen because of a patient or an individual shedding, could be transferred to another patient who could pick that up. So again, this is why with COVID you see all the extra cleaning going on of touch surfaces, so that minimizes the potential for this passive transmission through contact of those. When we look at vectors, vectors can be biological vectors or mechanical vectors. So with biological vectors, these vectors are actually infected. The pathogen is going to be inside of them. And so an example of that would be mosquitoes. So mosquitoes, right now, like Triple E is really big, but West Nile, those are actually inside of the mosquito. And so when the mosquito bites, it actually regurgitates a little bit into you. And so that's when we can have transmission of those pathogens that are inside of the biological vector. With a mechanical vector, it's a simple moving from one location to another. And so the pathogens are external. So the housefly, common fly, it actually has little hairs on its legs. So when it lands on a surface, it actually will pick up microbes that are present. And so when it moves to the next surface, it can, some of those will fall off, just like transient microbes. And so you can get spreading of illnesses that way. With diseases, we can have zoonotic diseases. Again, a topic that was a little more mysterious and unknown in previous semesters. But now you're probably aware of zoonotic diseases. These are diseases that originate in non-humans, but then get transmitted into humans. COVID-19 would be an example of a zoonotic disease where it was not originally in humans, but then made that jump into humans. And there's a number of them. It's increasing. And if we think about our interactions with animals, we have closer interactions with them than we used to. So rabies would be an example of a zoonotic disease. And so we know that vaccination rates of dogs against rabies, if the vaccination rate is high, we have less cases of human rabies. If it's low, we tend to have more cases of human rabies. And that's because of that close relationship that we have with dogs, right? And so the more interactions we have with animals, the more likely we'll see zoonotic diseases and increases of them. We're also sprawling. We're moving out into a lot of the habitat in which wildlife will live in. We tend to go on nature walks, right? And so all of these are potentials where we're interacting with these diseases, these microbes that would normally be in non-humans. And we, as we're walking on the trail, we might, run across some fecal material from an animal that has pathogens in it. Not that we're going and hopefully eating it, but just simply, if we accidentally get some on our shoe and then we're taking our shoes off later, we could potentially get some of that. So this table just shows some of the common zoonotic illnesses. So again, there's viruses, bacteria, but also there's parasitic worms like ringworm, tapeworms that occur. So a good number of those diseases, infections that you hear about in the news are zoonotic diseases. So with non-living reservoirs, again, these abiotic reservoirs, these can be environmental sources. It is important that you continue to interact with these environmental sources because they are a great source of microbes that are beneficial, because we are not water, we are not soil, so they don't tend to cause illness. There are definitely some soil microbes because of the production of endotoxins, and toxins could produce illness. But again, playing in dirt is a good thing. With diseases, we also have communicable diseases or non-communicable diseases. Communicable diseases are ones that can be transmitted from host to host, from human to human. So these would also be known as highly contagious agents, and that transmission can either be direct or indirect. So something like cold, the flu, COVID-19 would be communicable diseases. With non-communicable diseases, it's more of by accident that it gets transmitted. So that usually it's because of an interaction with a non-living reservoir that ends up causing illness. So tetanus would be an example of a non-communicable disease. If you had a friend that had tetanus and went and visited them in the hospital, the chances of you picking up tetanus from them would be extremely unlikely. But if you were, it's because of that interaction, because there's either been a like nail puncture, some kind of introduction of the Clostridium tetani to them, that they end up with that infection. Looking at patterns of transmission in communicable diseases, we can have direct transmission or indirect transmission. With direct transmission, we have to have close contact with an individual that has that. So this could be through kissing or sex, also exchange of droplets, so through talking. So again, 6 feet. Social distancing helps to decrease the chance of transmission when we have respiratory droplets being the route of transmission. We can also have vertical transmission. This is where it goes. Transmission is from mother to fetus through the placenta. And we can have these biological vectors through a bite that allows for transmission. For indirect, this is where vehicles are utilized. So fomites. So fomites would be non-living touch surfaces. And so things like door handles, the elevator button, the automatic door push button opener, those would be common kind of fomites. And again, that's why we see those being cleaned regularly with COVID. We can also have transmission through food, water, or biological products. And so sometimes this is where we get this fecal-oral contamination. So this is why it's important to wash your hands before preparing food. During the cooking process, a lot of times those microbes could be destroyed. But especially an issue if we have our produce that's not being cooked, like salad. This is where, transmission could occur. And so again, this is where making sure we wash our hands, making sure anything that's coming in contact with the food is dealt with. This semester I'm teaching a food microbiology course. In that food microbiology course, we talk about foodborne illnesses. We also talk about strategies to help to diminish foodborne illnesses. We also talk about fermented foods too. So it's a balance between good microbes and bad microbes. And then lastly, for indirect, we have where we have spreading through the air. So this is where the microbe can become airborne. And sometimes this is where there are very small droplets. So for direct, these droplets are larger. There is, the whole physics in this. There is a point where the droplets become so small that we would then consider that airborne. And again, this is where the big debate becomes with COVID-19 is how big do those droplets have to be? And so is it airborne or is it not airborne? And I think we're still not completely decided on that because there's definitely cases where the proximity of exposure between a positive individual and someone who ends up coming down with COVID did not justify there being it actually being respiratory droplets. It actually supports those droplets being small enough that it would be considered airborne. And this is why you should be covering your sneezes. So this gentleman is sneezing, and you can see all those respiratory droplets that are coming off of that sneeze. So you should always be sneezing and coughing into your elbow. And nowadays you should be wearing a mask so that sneeze or cough gets caught. And this is one of the reasons why the gaiters, if worn as a single layer, are not very effective, because a lot of this material can actually be dispersed and actually gets dispersed and thrown further if you have a gaiter on. Another type of infection is nosocomial infection. So these are commonly known as hospital-acquired infections. So individuals go to the hospital not just because they're sick, they go because they're healthy. And that may sound like weird, but yes, healthy people do go to the hospital, or people who are not dying. So people go to the hospital to have children, people go for medical appointments, follow-up care and such. And so if they end up developing infection while they're at the hospital, that would be considered a hospital-acquired infection. So there are between 2 to 4 million cases a year, so quite a variation there. There are epidemiologists at each hospital. They're supposed to keep track of hospital-acquired infections, but there's definitely people who, go to the hospital, get an infection, but don't come back to the hospital because the illness isn't severe. Maybe their PCP treats them, so they don't always get reported by the hospital. Definitely, it highlights the importance of aseptic technique. And with all these cases, it's a huge burden on our healthcare dollars. So there's a huge cost that goes with hospital-acquired infections. And so we definitely want to minimize the number of hospital-acquired infections as much as possible. The CDC has a website on hospital-acquired infections and also some of the stats. And you can also go to the Massachusetts website, Department of Public Health, and find out information on hospital-acquired infections, because both at the country, state level, all the information is kept. The most common source of hospital-acquired infections would be catheterization. So whenever we're introducing something from the outside into a patient, so for catheterizing them, we're inserting a tube into their urethra so that they can eliminate urine from their bladder. You have that potential, if it's not done aseptically, to introduce pathogens, microbes, into the bladder. Okay. Also, ports that are put in also are an area of hospital-acquired infections. So you should always make sure that you're taking precautions to prevent transmission of hospital-acquired infections, but also you potentially picking up an infection. And again, previously when we talked about, PPEs, personal protective equipment, that was kind of a, topic that seemed, what is this? But pretty much everyone knows what PPEs are nowadays. So making sure that you do take precautions to protect yourself. And again, usually this is in the hospital setting, but right now with the pandemic, not just in the hospital setting. So you always want to treat any patient, and again with the pandemic, I would say any individual, as potentially harboring infectious agents. So you don't know whether that person is asymptomatic or not. And so this is where making sure that to protect yourself and your family from ending up getting sick, that you do want to make sure that you abide by the social distancing and mask and hand washing and all those safety precautions that you can do to minimize the chance of infection. All right, so epidemiology is in chapter 13, but we're actually going to save that for later on. So I'm not going to cover epidemiology in this audio lecture. There will be an audio lecture in the future usually this fits into Unit 4 when we kind of do some miscellaneous topics. Epidemiology can be its whole course, so it kind of deserves its own lecture.